we present Molli, a new protein visualization tool. In this video, we will first demonstrate system features. We will then discuss the comparative and qualitative evaluations of Molli and conclude with results. Instead of console input, Molli employs mouse interaction for direct protein manipulations. Users may directly manipulate the structure, sequence, or residue representations. Currently, users may make alterations such as hiding and coloring residues, as well as changing the rendering style of the protein. Here's an example of a user using all three types of selection techniques. Additionally, Molli provides users simultaneous structural and sequence multiple alignment with the MAT structural alignment algorithm. Users may select one of three alignment views: overlap, synced in separate panes, and synced in different windows. To evaluate the effectiveness of the Molli system, a user study was performed comparing Molli's performance on directed tasks to JMOL. All participants were either biologists or computational biologists. Participants performed in tasks including selection, hiding, coloring, and changing the rendering style of choice proteins. The proteins used in the study were selected for increasing complexities: simple, complex, and very complex, based on the number of chains, chain patterns, and size. Here are some examples of directed tasks where each protein is of increasing complexity. Here's the task just viewed in Molli, now in JMOL. A measure of time to completion was recorded and used to compare the conditions. Both aggregate and by-group analyses were performed. Between conditions, aggregate analysis showed tasks 2, 5, and 9 are statistically significantly different in favor of the Molli interface. Those tasks included changing the rendering style of a simple protein, task 2, hiding selections on complex protein, task 5, and changing the rendering style of a very complex protein, task 9. Task 6, changing the color of a selection of a complex protein, shows weak statistical significance favoring Molli. Further analysis between conditions by user type, biologist and computational biologist, shows task 2 and 9 still favor Molli across types. Additionally, for the biologist user group, task 6 shows strong statistical significance favoring Molli, and task 4, a coloring task, favors JMOL, whereas the computational biologist group performed task 7, another coloring task, better using Molli.